So today I will be discussing about uh, the vacuole that is given in your syllabus together with the lysosome, and also I will try to finish off the microbodies which is also given. So let me discuss about the vacuole. Well, a vacuole, it is a membrane bound organelle. It is a membrane bound organelle that is present in different type of cells. Uh, in plant cells, we can see a large vacuole. In fungi, however, some protists, some animal cells, and bacteria also contain vacuoles, but in a very small amount. And most of the plant cells contain one or more membrane bound vacuole, right? So vacuole is found within the animal cells. It is a small membrane bound organelle, and that membrane we call them as tonoplast, inside which there is cell sap, okay, called as vacuole. So as I have said that it is a membrane bound organelle, and it is present in different types of cells including plant cells, including fungi, some protists, some animal cells, and bacteria also contain such type of vacuole. Immature plant cells contain several small vacuoles. Those plant cells which are immature, they contain very small amount of vacuoles, but in large number. But when the cell matures, a large central vacuole will be observed. So a large central vacuole will form as a result of fusion of these small vacuoles. So the small vacuoles, in case of immature plant cell, they will be fusing to form a large central vacuole in mature plant cells. The central vacuole consumes about 90% of the cell volume is occupied by the central vacuole in case of plant cells. Now this organelle that we are talking about, vacuole, it has no such basic shape or size. Therefore its structure varies according to the requirement of the cell, right? So again I repeat that this vacuole organelle has no such basic shape or size. Therefore its structure varies according to the requirement of the cell. And as I have said, tonoplast, it is the membrane that surrounds the vacuole organelle. Tonoplast is composed of 40% lipid, and it consists of 60% protein. Okay, so this is about the overview of vacuole. Now I will be discussing about the types of vacuole. There are different types of vacuole that we normally see. Okay, and like we can see gas vacuoles, we can see central vacuoles which are found in the plant cells, we can see your contractile vacuoles, food vacuoles. Different type of vacuoles are observed uh, on depending on the requirement of the cell, okay, as I have mentioned with you. So let me discuss about these different types of vacuoles. Okay, just wait for a minute. So the first that we will discuss is of about gas vacuoles. Gas vacuoles are also called as vesicles, and they are nano compartments which are freely permeable to gas. So in gas vacuoles, gases are freely can able to enter into these vacuoles, and this type of gas vacuoles occur mainly in blue green algae, also called as cyanobacteria, but they are also found in other bacterial species, in some archaea. In some archaea they are also being observed. Now, what is the function of gas vesicles or gas vacuoles is that gas vacuoles allow the bacteria to control their buoyancy. So, the main function of gas vacuole is to control the buoyancy of bacteria. Okay, and they are formed. These gas vesicles are formed when small bioconical structures grow to form spindles. The gas vacuoles walls are composed of a hydrophobic gas vesicle protein called as GPVA or gas vesicle protein A. Again, I repeat, if you are confused with this term, I am saying that these gas vacuoles are formed, that means they're composed, you can say, gas vesicle, the walls of the gas vesicles, the walls of the gas vesicle is made up of a hydrophobic gas vesicle protein A. So GPVA, also called as gas vesicle protein A, it forms a coating, it forms a wall around the gas vacuoles, and it is purely hydrophobic, not allowing the water to enter, right? Not allowing the water to enter. Now, this gas vesicle protein A, which forms the wall of the vesicle, it forms a cylindrical, hollow, proteinaceous structure that fills with gases. Now, this GPVA, or your, what can I say, the gas vesicle protein A, they actually forms a cylindrical hollow structure. They forms a cylindrical hollow proteinaceous structure. Proteinaceous structure. Now that structure is filled, that hollow structure is filled with gases. Okay. Now small variance in the amino acid sequence within the wall of these gas vacuoles, if there is small difference in the amino acid sequence, it produces change in the morphology of the gas vesicle. Okay, for example, GPVC or gas vesicle protein C, it is a larger protein. So again I repeat about the gas vesicles, not too much hard. Gas vesicles are also called as gas vacuoles. They are nano compartments which are freely permeable to gases. So gases can easily enter to these vesicles. Okay, and they are found mostly on cyanobacteria, but they are also found in archaea and some other bacterial species. Um Gas vesicles allow the bacteria to control their buoyancy, and these gas vesicles are formed, they are composed. The walls of the gas vesicle are made up of GPVA or gas vesicle protein A, which are purely hydrophobic in nature. This gas vesicle protein A forms a cylindrical hollow proteinaceous structure that will fill with gases. Now, if there is small variance in the amino acid sequence within these gas vesicle gas vesicle proteins, then it will change the morphology of the gas vesicle. Like there is also another type of gas vesicle protein called G GPV. Sorry, I have written GVP. GPV. GPV. C. Sorry, I have written again. I'm written mistake. It will be GVP. Okay, so it will be GVP. G for gas, V for vesicle, and P for protein. Okay, gas vesicle protein A and gas vesicle protein C. Okay, now we will discuss one another type of vacuoles which is called as central vacuoles. So the central vacuole are those vacuoles which are found mostly on plant cells. Most mature plant cells have one large vacuole which typically occupies more than 30% of the cell volume and can occupy as much as 80% of the volume for certain cell types and conditions. So, central vacuoles are mostly observed in the plant cells where from 30% to about more than 80% your it can occupy within the plant cells volume, and strands of cytoplasm are often run through this vacuole. Now this central vacuole are surrounded by a membrane. It is surrounded by a membrane, so there is a membrane which forms around the central vacuole called as tonoplast, as I have said before. Also, it is known as the vacuole membrane, vacuolar membrane. It is also known as vacuolar membrane. Okay, so as a membrane, it is mainly involved, tonoplast is mainly involved in regulating the movement of ions around the cell and isolating the materials that might be harmful or threat to the cell. So, the function of this vacuolar membrane is to regulate the movement of ions, regulate the ion movement across the cell, around the cell, and isolating the materials that might be harmful or threat to the cell. Now, transport of protons from the cytosol to the vacuole will stabilize the pH, will stabilize the cytoplasmic pH while making the vacuolar interior more acidic, creating a proton motive force which the cell So as I have said here, what I have said here that wait. So I have said here that the transport of protons from the cytosol to the central vacuole will stabilize the pH, will stabilize the cytoplasmic pH while making the interior of the vacuole more acidic, and thus it will create a proton motive force by which a cell can use to transport the nutrients into or out of the vacuole. So when suppose if this is the central vacuole, if this is the central vacuole, okay, suppose, and around which this is the cytosol, due to transport of the protons from cytosol to the vacuole, it stabilizes the pH in the cytosol. The pH is around is 7, we know 7.2 something, and making the central vacuole more acidic. So there will be a acidic pH inside the central vacuole, as we have seen in lysosomes also, and there it will there due to this it will create a proton motive force. Now what is a proton motive force? Proton motive force is the movement of ions. It is that force that is created due to electrical potential gradient, like cations, for example protons, the H plus ions, okay, or anions also can be responsible for this type of force, for this type of electrostatic force. So due to the occurrence of this proton motive force, what happened? Due to occurrence of this proton motive force, it will help, that force will help to transport that nutrients into or out of the vacuole, okay, and thus your the function of the vacuole can be done. Although a single large vacuole are most common, the size and number of these central vacuoles can vary in different tissues and in different stages of development, right? So as I have said that this central vacuole can vary depending on what type of tissues are there, at what developmental stage is present, is actually is running through that phase is running through that plant. Depending on that, central vacuole can vary, like in meristems. Meristems contain small provacuoles. They contain small provacuoles, and the cells of vascular cambium may have small vacuoles in the winter and one large one in the summer. So meristems have small provacuoles, while the cells of vascular cambium, well, I think you know about the vascular cambium cells, tissues. Vascular cambium is the main growth tissue found in the stems and roots of many plants. Specifically in the dicotyledonous plants we can find, right? In the gymnosperms we can also find this type of vascular cambium tissue, like in pine trees and other vascular plants, that means those who have xylem and phloem vesicles, right? Now, normally, this vascular cambium present between the xylem and primary xylem and primary phloem. Okay, so as I have said that this type of vacuoles can vary. It can vary. Okay. Now, aside from storage, the main role of this central vacuole is to create a turgor pressure, to maintain a turgor pressure against the cell wall. So, the main function of your central vacuole, first I have said the main function is storage and your transport of nutrients into or out of the vacuole. But another function is to maintain the turgor pressure, maintain the turgor pressure. Now, what is turgor pressure? Turgor pressure, it is the force within the cell that pushes the plasma membrane against the cell wall. Again, I repeat, turgor pressure, also called as hydrostatic pressure. This pressure is the force that occurs within the cell that pushes the plasma membrane against the cell wall. Okay, it is caused by due to the osmotic flow of the water that occurs in here in plants. It also occurs in fungi as well as in bacteria. Okay, so the central vacuoles also play an important role to maintain the turgor pressure right against the cell wall. Now the proteins which are found on the tonoplast. So the tonoplast is the membrane which surrounds the central vacuole. So the proteins which are found within the tonoplast such as aquaporins, they control the flow of water into and out of the vacuole through the help of active transport. Again, I repeat, the proteins which are found in the tonoplast, such as aquaporins, they control the flow of water into and out of the vacuole through the help of active transport. So aquaporins are actually here control the flow of water into and out of the vacuole through the help of active transport, and they also pump the potassium ions into and out of the vacuolar interior in this way, in this process. Okay, now due to osmosis the water will diffuse into the vacuole and placing a pressure on the cell wall. If water loss leads to significant decline in the turgor pressure, the cell will, as a result, it will carry out the plasmolysis or plasmolyze. So again I repeat. So as I have said that the proteins which are found in the tonoplast, the proteins which are found in the tonoplast like aquaporins, they control the flow of water into and out of the vacuole through the help of active transport by pumping the potassium ions into and out of the vacuolar central vacuolar interior of the vacuoles. Now Due to osmosis, the water will diffuse into the vacuole. It will diffuse into the vacuole and it will place a pressure on the cell wall. Now, if water loss leads to significant decline in the turgor pressure, then it will create plasmolysis or the cell will plasmolyze. And turgor pressure exerted by vacuoles is also required for cellular elongation. The pressure which is done by the central vacuole, it creates the turgor pressure which actually makes the cell to be elongate. As the cell wall is partially degraded by the actions of expansins, the less rigid cell wall is expanded by the pressure coming from within the vacuole. So partially the cell wall of the plant is degraded by some proteins called expansins. That's why due to interior pressure done by the central vacuole, it, it gets elongated. Okay. So the two function I have said, storage and maintain the target pressure. Another function of the central vacuole is that it pushes all contents of the cell cytoplasm against the cellular membrane and thus it keeps the chloroplast closer to the light. Again I repeat, I am erasing this part. So another function of the central vacuole is it pushes all the contents of the cell cytoplasm against the cellular membrane. It pushes all contents of the cell cytoplasm all contents of the cell cytoplasm against the cell membrane against the cell membrane, and thus it keeps the chloroplast closer to the light. Most plants store chemicals in the central vacuole that react with the chemicals in the cytosol. If the cell is broken, for example, by a herbivore animal, it is eating the plants, then what happen? The two chemicals can react to form a toxic chemicals. Again, I repeat. So most plants store some chemicals within the central vacuole. Now that chemicals can react with the substances which are found in the cytosol. Like for example, if a herbivore animal like cow or cattle is eating some plants and cause injury to the plant cells, obviously it will call injury because they are eating. So that two chemicals from the vacuoles and from the cytosol will react to form a toxic chemicals. Okay. Like for example, the best example is what? The best example is in case of onions. Well, when we cut the onions, an awful gas comes out which causes us tear formation. We tear, our eyes started to water. It's because of the production of syn propanethyl propanethyl syn propan propanethyl propanethyl S oxide. So due to the production of this chemical as a result of reactions caused by the central vacuole, The chemicals found in the central vacuole react with the chemicals found in the cytoplasm to produce this type of syn propane ethyl S oxide, which causes us to create a toxic substance that causes us that makes a awful smell and our tears starts to come out. Similarly, we find in case of garlic also. In garlic, aniline, aline and the enzyme alinase are normally separated but form allicin if the vacuole is broken. So aline, again I repeat. So aline and an enzyme called alinase, they are separately found within the vacuole and in the cytosol, but when we cut them, they get mixed together and alinase break down the aline to form your allicin. That's why we feel a pungent smell in the garlic when we cut. Okay, so as I have said that different functions done by the central vacuole. The first function I have said is what? I have said first function that it is mainly involved in mainly regulating the movement of ions around the cell and isolating the materials that might be harmful or threat to the cell. Then it also stabilizes the cytoplasmic pH. Okay, it maintains the, as I have said, the target pressure. Okay, and against the cell wall. And I have also said that it also pushes, the central vacuole also pushes all the contents of the cell cytoplasm against the cellular membrane, which as a result causes the chloroplast to keep close to the light. And some chemicals are also stored in the vacuoles that can react with the cytosol if that chemicals get exposed out to the cytosol. Like for example, allicin I have said. Okay. Now the vacuoles, which the central vacuoles which are found in the fungal cell, the central vacuoles which are found in the fungal cell perform similar functions as those in the plant cells, and there can be more than one vacuole per cell within the fungus. In the yeast cell, the vacuole is a dynamic structure that can rapidly modify its morphology. Now, these vacuoles which are found in the fungi, which are found in the yeast, they perform many processes like homeostasis. They perform homeostasis of the cell's pH, meaning the pH and concentration of ions. They also carry out osmoregulation, storing the amino acids and polyphosphate, and degradative process, as we have seen in lysosome. So again I repeat, so the vacuoles, the central vacuoles which are found in the fungi and yeast, they are mainly involved in homeostasis of the pH of the cell's pH. Then to maintain the concentration of ions. Then they store the amino acids and polyphosphate, and they also carry out the degradative process as we have seen in lysosome. Some toxic ions are also um, transported into the vacuole to separate from the rest of the cell, and those toxic substances are strontium, which are designated by SR 2 plus, cobalt CO2 plus, and lead. Well, lead is a heavy metal that you know. So these toxic ions are also stored in the stored in the central vacuole and separate from the rest of the cell. Okay, so this was about the central vacuole. Now we will talk about two more vacuoles. One is your contractile vacuole and another one is your food vacuole. So let us see about the contractile vacuole. Well, your contractile vacuoles is a specialized osmoregulatory organelle that is present in mainly free living protists. All the protist groups, free living protist groups, have this type of contractile vacuoles. Okay, so they are found in free living protists. Now, the contractile vacuole is a part of the contractile vacuole complex, which includes your radial arms and spongiome. So, it consists of the contractile vacuole actually consists of what? Radial arms, and it also consists of spongiome. Spongiome. Now, what is actually spongiome? Spongiome, plural, it is called as spongiomes. It is a contractile, it is a structure which surrounds the contractile vacuole. Okay, so spongiome is what? Spongiome actually it is a part which surrounds the contractile vacuole, and this spongiome perform many functions. Okay, like transport of water, like transport of your water into the contractile vacuole, writing a CV here, and in also helps in localization and docking of the contractile vacuole. Okay, I hope you have understood about the spongiome. Now this contractile vacuole works periodically, contracts to remove the excess water and ions from the cell to balance the water flow within the cell. So again I repeat, the contractile vacuole normally functions to remove that excess water and ions from the cell, and it actually balances the water flow into the cell. So the main function of contractile vacuole is to balance. You can write remove excess water and ions from the cell, from the cell, and it also may balances the water flow into the cell. Now when the contractile vacuole is slowly taking the water in, obviously the contractile vacuole become enlarged. Again I repeat, so it is saying, so what I have said of the function that contractile vacuole, what they do, it remove excess water and ions from the cell Why? Now to balance the water flow, to balance the water flow into the cell, into the cell. Now when the contractile vacuole is slowly taking the water inside, what will happen? It will become enlarged. Now this enlarged contractile vacuole is now called as diastole. Diastole. Okay, don't get confused with the heart diastole, systole diastole. So when the contractile vacuole is slowly, it is slowly taking the water. So this is contractile vacuole. I'm writing a CV. Now it is called as diastole. It is slowly intaking the water inside, and the contractile vacuole enlarges. So it become enlarged. It becomes big in size now. Now this is called as diastole. And when it reaches its threshold, that means up after that it will not able to take the water. Now the central vacuole will contracts. The contractile vacuole will contracts, which forms systole to release that water. So after the threshold value is reached, it will now start to release that water. Now it will call as systole. Then we will not call as them as diastole. We will call them as systole at that time. So when contractile vacuole is slowly taking the water in, the contractile vacuole will become enlarged, and we call them as diastole. After reaching to a threshold value, then it will starts to release that water. Then we will call them as systole, periodically to release the water. The last part is your food vacuoles. Well, food vacuoles, as the name suggests, it has only one purpose. Food vacuoles, also called as digestive vacuoles, also called as digestive vacuoles. They are mainly found, they are the organelles which are found in ciliates, which are found in Plasmodium falciparum. They are found in Plasmodium falciparum, and they are also found in ciliates. Ciliates are the group of protozoa. Ciliates are the group of protozoa that are characterized presence of a hair-like structure, organelles called cilia. Okay, which are identical with the flagella found in the eukaryotic cells, but they are much shorter. Okay, they are much shorter as found in the protists, as found in the protozoans. Okay, so they are found in this type of cells, the food vacuoles. And as we know, Plasmodium falciparum, what it do? It is it is actually Plasmodium falciparum. What they do? They do disease. They are the vectors. They are actually found within the mosquito called Anopheles mosquito. Anopheles mosquito to cause you malaria. Okay, so they are found in these organisms, these food vacuoles, also called as digestive vacuoles, found within the ciliates. Found those who are having cilia are called ciliates, all the protozoans. Then it is also found in the Plasmodium falciparum, which is a protozoa which cause you malaria. Okay. Now we will discuss about the function of your central vacuole. Just like lysosomes, the function of vacuole is somewhat similar. First function is vacuoles contain a variety of hydrolytic enzymes. Vacuoles contain variety of hydrolytic enzymes, and in addition with the hydrolytic enzymes, plant vacuoles also contain inhibitors of serine endopeptidase. Second function is, particularly in the case of plant vacuole, the central vacuole, is the solute accumulation. Solute accumulation within the vacuoles. Well, the solute accumulation like your chlorine, like your potassium, like your sodium, then some amino acids, some sugars, okay, some organic acids like malate, organic acids like malate, citrate, and secondary metabolites are accumulated by the vacuole. Vacuole synthesizes some of these molecules also, but mostly they are imported from cytoplasm. The third function of the vacuole is the presence of the internal acidic environment leads to degradation of the large molecules sent to the vacuole for destruction. Okay, so they cause destruction also because of this acidic environment, and they also leads to a proton motive force also. I have said before that the interior of the vacuole is more acidic, that creates a proton motive force which causes the cell to transport the nutrients into or out of the vacuole. Okay, and due to the presence of low pH, the degradative enzymes also acting on the vacuole, so they cause degradation of macromolecules. I'm writing as here. They cause degradation of the macromolecules, right? There are also other function. Vacuoles also remove potentially toxic substance from the cytosol, like your heavy metals, like your herbicides. So another function is removal of toxic materials. Removal of toxic materials, as I have mentioned with you before, that some toxic ions like strontium represented as Sr plus 2, then cobalt, Co plus 2, lead, which is a type of heavy metal, they are separated by allowing its entry into the vacuole and isolate them from the rest of the cell. Okay, so that no such harmful effects occur by the action of these ions, by the action of these chemicals. Okay, then they also help in lysis and recycling of the misfold proteins that have begun to build up within the cell. So the misfold proteins, that means those proteins which misfold proteins, the misfold proteins which are not actually folded properly, and misfolded proteins are non-functional or they can cause abnormality also. Okay, so it has to be removed out from the cell. So vacuoles serve to help in lysis of those misfold proteins. They have hydrolytic enzymes also. They have that acidic pH, ideal environment to cause the degradation of this type of misfold proteins, and this is what the function of vacuoles are. Okay, Some more functions are also there present within the vacuole that we can write like for example isolating the materials that might be harmful or threat to the cell. Then it contains all the waste products. It contains the water in the plant cells, maintaining the target pressure, maintain the internal hydrostatic pressure or target pressure within the cell. It maintains the acidic pH inside the vacuoles, just like lysosomes. Okay, it also export unwanted substance from the cell, and especially in plants it allows the plants to support structures like leaves, like flowers, due to the pressure by the central vacuole. Okay, now there in seeds also we can see some modified vacuoles where the proteins are also stored and are called as protein bodies. So within the seeds vacuoles are also found, but those vacuoles stored the proteins and called as protein bodies. Protein bodies. Okay, which are modified vacuoles. So these are the function of the vacuoles. Well, other functions, many functions. You can mix with the function with the lysosomes also. That like it also involve in autophagy, digestion of its own internal organelles of the cell. Okay, and degradation of many substances and cell structures in the certain organisms. Like I have said now that misfolded proteins can be digested. So it is a type of autophagy, maintaining that environment. Okay. So this was all about the structure and function of your vacuoles. I hope you have understood what I have discussed with you. Um, just watch this video lecture. I'm requesting you again because in this way I'm taking your classes, and I will give you the notes. Generally you can watch the notes very wellly. You can go for Verma Agarwal book, okay, that I have already said, and you can also you can also search in them in this vacuole structure and function in Wikipedia. It's also beautifully it has been mentioned here there. Okay, so that's all for today's lecture.